Yes. Good evening. Good evening, Jason. Mr. Jason, good evening.
Venkat.
Yes. How are you, Jason?
Fine, thank you. How are you?
Yeah, it's fine. Yes. Good evening, ladies and gentlemen. This is Dr. P.V. Rao, St. Mary's College of Pharmacy Principal from Guntur. So today I am very much happy for this online webinar, role of bioinformatics in pharmaceutical sciences. So in this regard, I am very much thankful to our chairman, sir, Dr. Reverend K.V.K. Rao Garu, and my managing director, sir, Sri V.V. Ratnakar Reddy Garu, campus in charge, Mr. Y. Raghav Garu, and all my faculty, teaching fraternity, and my dear students. And I am very much thankful to my friend, Mr. Uh, Jason, Benjamin Jason. So he is my friend and he is my colleague also. Thank you very much. So beyond this hectic schedule also, you came forward to nourish the students about this bioinformatics. Thank you very much, Jason. So my faculty, Miss Shantipriya, will address about this gathering and your introductory remarks of your biodata. So please proceed, Shantipriya.
Okay, sir. Hello, very good evening to one and all. So this is Shanti Priya on behalf of St. Mary's College of Pharmacy. So St. Mary's Group of Institutions, Guntur. I coordinately invite you second international webinar on topic role of bioinformatics in pharmaceutical sciences. I am very happy to introduce today's eminent speaker and dynamic personality, Professor UNDETY Benjamin Jason Garu. A profile regarding the speaker, Professor Benjamin Jason Garu. So he worked as a pharmacist in Care Hospital, Hyderabad, and after that he worked as lecturer in Bharat University at Chennai from 2006 to 2007. So he worked as lecturer in Satyabama University at Chennai from 2007 to 2015, up to 8 years. He worked as production operations manager in Apotex Incorporation at Toronto, Canada. Then right now, Professor Benjamin Jason Garu, working as a senior formulation and technical assistant at Thermal Fisher Scientific Incorporation, Canada. And also he working as a professor part-time Durham College, Canada. So SAW has many international and national publications, published research articles, papers in different journals, and SAR organized many workshops and seminars during total in teaching experience 10 years. So thank you for accepting our invitation to ignite the students with your knowledge, sir. So now I request to Professor Jason Garu, please start your eminent sessions.
Thank you, Shanti Priya. Thank you, and Venkat.
Yes, yes. I have given this one, sir, just your slide.
Can I go
Yes.
slide 1?
Yeah. Now it is topic covers like that.
Oh, okay. Yeah.
Yes, you can start.
Okay. Good evening, students and all faculty members of St. Mary's and maybe, I don't know, other colleges. And it's my pleasure and honor to be part of this session. Yes, it's an opportunity to talk again after a long time to students and to the academic fraternity. I've been in teaching for a while, almost now 5 years, close to 5 years. Think this much, not teaching students. So it's my pleasure now, and I enjoy teaching. But today's session is not like I'm not presenting anything of research. I'm not going to scare you, students. And maybe for some faculties, this may not be new. Or topic I'm actually focusing to students mostly, so that you guys can think, and if something is enlightening you or igniting you, you can think about it and try, like, you can try to integrate, like, wherever you feel, maybe in your academics or in your projects or maybe in your as your career. So bachelor's in pharmacy, I took master's in bioinformatics, and I was teaching almost 9 years of bioinformatics to BTech and MTech students. So coming from pharmaceutical background, bachelor's and little bit of pharmacy experience, and now after coming to Canada, working in pharmaceutical companies, and with my master's in bioinformatics, teaching 9 years, I see like there's a very good combination of these two streams. And bioinformatics as such is as it's an interdisciplinary combination of different other sciences. So I myself, like, I'm a combination of these two, bioinformatics and pharmacy. So I'll try to give a glimpse where you can think what you're studying and like what you are planning, or if you don't have a plan, you can start what you can do with bioinformatics, or what you need not do if you're not interested. Okay, today I'm going to cover topics, closing my cam, and we'll just go proceed with the slides. Uh, I don't know whether I'm audible, because this is the very first time I'm talking to audience whom I don't see. Uh, Maybe at least one or two. Anyone who is watching, can you just chat? Yes, I'm audible. I can hear you. Your my voice is clear. Whether I'm understandable, am I speed or slow? If anyone is there, maybe you can ping a message whenever you are ready. Okay, today I'm going to give like cover topics, introduction of bioinformatics, like what it is and what's the history and so on, and where, what kind of jobs or like what kind of career the bioinformatics provides, and little bit of branches. There are a lot of, it's a wide area bioinformatics, but you, and maybe we'll discuss one or two topics which is related to pharmaceutical sciences. And then chem informatics, it's also one of the branch actually. And you have databases, software, uh, uh, resources, and then where exactly bioinformatics is applied. Uh, Hi, Venkat. How can I go to the next slide?
Yes, you can go in as your desktop on.
Okay, okay.
Yeah, you got introduction.
Uh, okay, okay.
Yes,
Hey, you got it? Okay. Yeah.
yeah, sure, nice.
Sorry, guys. uh, this is completely a new thing for me. I'm not very much I mean, don't have any experience presenting anything in online like this. But anyways, yeah. So, introduction. What is mathematics? What is the definition of it? It is a science of collecting, classifying, storing, analyzing large and complex biological and biochemical data using computers. So, as the word bio, what is bio? Bio means life. Informatics. Informatics is coming from IT. IT comes nothing but computers, information technology. So, this is a fusion of IT with bio. And when it talks biological data, and we also have biochemical data and even just chemical data. Maybe I can redo the definition because today we're going to talk about both things, bio and chemical, otherwise meaningless, because I know you're all from pharmaceutical background, like, or you're studying pharmaceutical, bachelor's and master's, and maybe you would try to venture into this line if you wish. So, bioinformatics, actually, it started, the history of bioinformatics is like, it just didn't evolve as such, but in the course of time, decades of data being generated through wet labs and different experiments and research, whatever data was generating on a daily basis, it has become so massive. The papers can't contain, and it's not only just the problem containment, uh, uh, it's also like how you retrieve the data, extract the data what you want. So then it is then when the computers are invented or designed, and the data storage concept has evolved, and then people started utilizing this opportunity and they started storing it. So, say for example, the sequence data of a protein, the very first protein, uh, the first protein in sequence. So, like that, you have a lot of sequence being uh, sequenced, maybe nucleotides or proteins, and then even they started getting the structures. All these things can be stored. It's not only just storing, you can also do the analysis. You can retrieve and you can look for the data what you wanted. So this is a little bit of history of mathematics. And then there are two major areas like in mathematics. One, two. The number one is bioinformaticists who develops a software or a tool or a database or whatever it is, and they use programming languages. Mostly in bioinformatics, it takes mostly the scripting language. It's very simple. The syntax is very simple and abstract. People use mostly Python all these days, but now they started using, sorry, they were using Perl before, and even now they use, but now using Python more and Java and R. and relational database, and so on. So bioinformatics is nothing but something like pharmacists who uh, produce compound medicine, right? So bioinformatics are the who develop, but bioinformatician is on the other end where we all fall in. But if you're a good programmer, you can also do both. So bioinformatician is the one who's using the software or a database or any kind of program or an algorithm which is being developed and implemented for solving problems or any kind of research questions or like whatever kind of projects you're. So that is what it is like. Two areas: bioinformatics and bioinformaticians. So now the branches in bioinformatics. Uh, I'm just checking if anyone has given any, but I don't see anyone giving any comment. That's fine. I assume that what here and you're able to understand. Any questions? You can make use of the private chat or oh, sorry. Uh, I'm seeing some comments. Yeah. Yes. I can see. Oh, thank you. Pharma classes. I think it's a group, not an individual. How to apply jobs? I'll be there. Uh, yeah. Good morning for me. Anyway, thank you. Okay. Let's go back. So the branches in bioinformatics. As I said, it started with sequencing and structure, mostly with biological systems. So you have first two things, genomics and proteomics. Genomics starts with like working with genome, but the complete sequence, DNA sequence. And gene is something like a part of genome where it can express, okay, expression or it can show its function. So when it is expressing and when it is showing its function, it codes amino acids. Amino acids, the moment they are uh, the expression, they form a chain with a peptide bond, and the moment they formed a chain, instantaneously they form a structure. It's called a protein structure. So genomics, proteomics. Uh, so there is a large scope, like it's a huge area where people work on just sequencing DNA or just sequencing a protein, and also working the structural part of genomics and structural part of protein, and then functional parts of both. So all this comes under genomics and proteomics. And when it comes to expression, uh, I didn't add this here, but the central dogma of molecular biology is you have to start from DNA, then you get RNA transcription. From RNA, it translates into a protein or a chain of amino acids called translation. So there is a study of just transcription. So it's working with messenger RNA expression levels, and then you have cheminformatics and medical or health informatics. So if any questions, explain a little bit faster. Okay, thank you for the comment. That's good. So I'll go a little fast now. Cheminformatics, as the word indicates, it's just with chemistry. So you're going to work with chemical concepts. Until now, the first three topics, it's more like molecules. They are much complex. They're huge. Their uh, size is so huge. The coordinates and the data, the storage is so huge. But coming to chem informatics, the molecules are very small,
The
and
data, the
only
storage is so
molecule
huge. But coming to chem informatics, the molecules are very small. The data, the storage is so huge. But coming to chem
or
informatics, the
I'm hitting
molecules are very
myself.
small. The data,
So here, yeah, we are doing searching these uh, small chemical or the molecules which are across the database and clustering them and quality structure activity relationship. Okay, we'll talk about it later. And virtual screening. And the next one is medical and health informatics. It's more of patient healthcare information. How it is applied to us? Very much applied to us because pharmacy is a part in a hospital setting, and we have hospital pharmacists. They to play vital role, but you also have hospital bioinformaticians or bioinformaticists who work straight from the patient data. And maybe I can talk a little bit about this when we come to the of that related topic. Okay, now drug design and discovery. Conventionally speaking, like even now we have the same method uh, where bioinformatics doesn't play a role. That is what I mean to say, conventional. So just by the picture, you can understand like how many compounds to screen. Here it is 10,000 to 20,000. There is no end. And you optimize, get a lead and optimize it, and which is the most probable candidate. And then you start candidate. You do lab studies or like in vivo, in vitro, preclinical and clinical, and then altogether it takes minimum of 10 years, and it can go even further. And how many billions of dollars is being invested? So bioinformatics, when it comes like when you see this chart, uh, bioinformatics will come with the very first stage um, of this flow chart, where you need not screen thousands and millions of compounds in vivo, in vitro, or in vivo, whatever it is. So everything can be done in silico in a computer, where you have an environment working both with the target and your most probable lead molecule. Okay, so this is most of the time we spend, I mean, large amount, 10 to 12 years is spent at the very first point. And even it is spread across all other levels. So now coming to the bioinformatics, like modeling and drug design. Modeling is something which is both with the and your small molecule. So as I mentioned here, the receptor based, like there are two types of modeling a drug, um, which is receptor based and ligand based. So the next you can see the first part is based on the receptor, and the second part is based on the ligand or the most probable drug molecule. So when we say a target based or the structure based, it is nothing but a protein. Mostly, it's a protein it is a functional part of or the function where the DNA or RNA is expressing, right? So this is the place where we are targeting, where our lead or drug molecule is going to be fused, or it is going to bind, and we get our desired biological actions. So biological action is nothing but arresting the disease, arresting the progression of a disease.
So
Or
while it actually is nothing but
so
arresting
first part is the structure based
the
ligand. So here let's say our target structure is the protein, and so first part is like as a bioinformatician modeling a structure of a protein. Little later, how many database like kinds of databases are there and what types of databases are there. There you can see like uh, how many protein sequences are there, but how many protein structures is there. So still there is a lot of work to be done to get the structures of all the proteins. Uh, but this is a good area where people can work getting structures, whether in vitro or in silico. So this is talking about in silico with already having amount and large amount of protein sequences. We can elucidate or build a structure from the existing sequences. And there you come with sequence analysis, getting the most similar motifs or the functional parts of that particular existing structures of those existing sequences. And anyways, the structure of a target molecule has been designed, and then uh, the next job in target receptor based drug designing is like getting the most possible active site. Active site is nothing but the place where our ligand is going to bind. So based on that active site or the based on that most functional part where you have a larger cavity where you have most possible interaction can happen. So that's a place we can either design a ligand or screen this particular site across it and get the most possible sites. So that is what about designing. Ligand docking is nothing but once you get this, you dock with your functional part of the protein structure or the cavity or the active site, and then see how the scope, what kind of energy, and working with molecular dynamics or molecular mechanics, and seeing um, which is the most possible uh, molecule for our structure. And then pharmacophore modeling. Pharmacophore is nothing part of our own lead molecule, working with different functional groups, which is best one, getting best docking scores and so on. And the next part is ligand-based computer-aided drug designing. So it is working with ligand alone. So ligand structure, you have databases, you have a lot of information already, lot of structures already. It's a huge area collection you can work, download, and then based on the node structure, whatever you have already designed or whatever you already want, you can do the structural and quality structural activity relationships, or you can model different other pharmacophores with the ligand what you want, and then or you can also screen across the ligand database. So it's then you get a lot of leads and you can get some most closest hits of the database, and then at last come down to the most probable lead, and that lead you're going to optimize, and that can be the most potential drug candidate. uh, just checking if there is any questions. Okay. Now, this is what I already spoke, showing you this flowchart. So, the receptor-based drug design, you have the receptor, usually a protein, and then you're going to determine its structure. The central dogma, what I said, from DNA to RNA, RNA to protein. First part is transcription, and then the second part, RNA to protein, is your translation. So the structure is the protein itself is nothing but structure. DNA and RNA you have in sequences. The moment you see a protein, you don't have it. You have a sequence, but it doesn't stay stay linear. It always falls into a three-dimensional structure. So that is what we want for our drug design. So yeah, drug molecule binds to cause biological effects. That is what we expect, but very good, effective biological actions we need. And lock-in key system is nothing but which see where our drug molecule binds to the active site of the receptor. It is just like a lock-in key system. So it is even more than that actually. Once it binds, it can't leave it. And it do its work and it goes on. And high throughput virtual screening protein targets are screened against small molecules or small compounds to see which molecules bind strongly to the target. And this is the schematic diagram, like how it goes. I already told verbally, but yeah, structure and then building a model for the binding site of that structure of protein and searching the database for the new candidates and docking it, predicting the binding constants or seeing the activity, and then since getting like most probable drug molecule, and then it comes to us as a pharmacist or a pharmaceutical technician or like maybe B form or M form, synthesizing that particular molecule. And ligand uh, based drug design. Ligand is a molecule that binds to another molecule. Drugs are considered to bind receptors, and any chemicals that bind to receptors are usually termed ligands. Ligand which can bind. And you might be confused. Like I'm using a lot of terms here: ligands, leads, and drug candidates. So ligand is nothing but it's just meaning is which can bind. Lead is something like the most potential candidate or a compound which can lead to be a drug. Okay, and drug candidate, uh, it cannot be a drug molecule until you finish the clinical trials. Actually, uh, it is not just bioinformatics alone, but it has to finish all the trials and then pass the trials and be a drug molecule. So until then, you can say it is a lead compound or a most potential drug compound. So you also have other varieties, like you can take the existing drugs and you can modify the functional groups, which is already there in the market, but it can be very easy in silico working with the functional groups. As I said, the pharmacophore modeling, it's both existing drugs or with the natural products, where you have a subject called pharmacognosy, where you have the natural extracts or the most active ingredients and get their structures and see that they are effective, or you work with their functional groups, the pharmacophore modeling, design and see and get the most uh, effective therapeutic values, and then you can do other things once you finish this. So I hope you guys understood thus far. Thank you, Deepika. I have a habit of calling my students and talking. Don't get embarrassed that I'm calling your name. Uh, Now, this is the schematic database for designing uh, database, or you model it, and then you have a receptor, the structure of a protein. It's huge. According to that, you have all those uh, atoms and the interaction between them. And on the right side, you have the ligands. They are small molecules. They they dock with are on a particular site, and then that is how the protein is denatured. So it is called actually denaturing of a protein. The nature of the protein is being lost. That is our actually main aim when we are working anything. Maybe just for example, cancer. So our aim is to dock with that particular type of disease. Okay, now the advantages. Yeah, it is very uh, time saving. The first thing you need to spend a lot of time. It automatically reduces your cost and very fast. Automatic. You're not actually working, sweating, working with a lot of in vivo instruments. Because like in pharmaceutical industry, I can understand back to the clinical trial batch or a commercial batch. There's a lot of cleaning, lot of there's a lot to do in the actual process, but automated process takes physically, even financially, or with time. And then you can obtain an idea about drug receptor interaction. Just go to the level of in vivo, say for example in animals or even in human trials. You'll have an idea like what kind of output is there when you have a drug and receptor interaction. Minimize lot of you can also study a lot of toxicity and minimize the synthetic and biological testing efforts. As I said, even your testing too many things, testing process itself will be reduced, and minimize the synthesis of it. Once the lead molecule is designed, you have a structure. Based on the structure and based on the bonds and based on like the compounds involved, as a pharmacist or as a chemist, you can design like the uh, of synthesis of this particular candidate. So and then you have minimized chances for failures in the final. Yes, of course. Not in spite of spending years and billions of dollars, there is a chance like it can fail, and it is most common. We see pharmaceutical industry failing uh, phase, and you can also like uh, can anyone name any drug which is being withdrawn from our market? Can you put a comment? Appreciation, but can somebody answer like uh, no? Okay. There are some drugs which are um, also being studied uh, after being launched into the market. It's called post-market study. Yes, hydroxychloroquine, పెదరాయుడు. Thank you. Yeah. So even then, there is a chance, or there are some drugs which are being withdrawn. So that can be minimized. And even before it goes to that far, it can be minimized. And I forgot to add one more point: the advantages of computer-aided drug designing. I think I wish to go back to the very first slide. Sorry, yeah, here in the third, the last one, medical and health informatics. So patient care information, or uh, exactly where the patients come, the hospital. So I don't know how far it has already started in India, but here I see in Canada, hospitals, they need a lot of bioinformaticians. You can also Google and Indeed. Somebody asked like job aspects. You can Google. Students, the best place for you, you have Google. Google whatever you want. So I just want you to Google Indeed.ca in Canada and see hospital bioinformaticians or hospital uh, informaticians. So people work when a patient comes, they take the sample of a blood, and the patient's DNA or the patient's particular disease causing expressing gene is being sequenced and structured, or like in doing any kind of analysis based on the patient sample. Once you get the patient's target molecule, they're going either gene therapy or any particular most potential drug which can be perfectly suiting to that particular patient can be diagnosed. So here it also comes with another concept called personalized drug therapy. Maybe some of you have heard this. Maybe those who don't know, I just want to elaborate a little bit more. Personalized drug therapy is nothing but what we have now currently is not personalized. It is like one drug uh, designed to everyone. But personalized drug therapy is nothing but a drug molecule tailored to an individual. So, like now we have all the clothes which are ready made, but the days, even in my days, like my friend Venkat and maybe some faculties, uh, we stitch our clothes tailored to us, which perfectly fits, but the stitching quality may not be good. So tailoring a drug to a particular patient is you get a most precise medicine. So there you come a concept called uh, precision medicine. Okay, precision. What do you mean by precision? It is very precise. It's exact fit. So you don't have a scope of just trying a chances, but fitting exact to the target, and you get the desired effects. Sorry. So okay, this is where we stopped and went back to that. So that is one point I want to add it here. Advantages of CAD is that's the future, and I was just going through when I preparing these slides for you guys. Uh, all these slides I just prepared within 2 days. Still, I feel that I don't have much information to you, but you can, as I said, all everything is in front of you. Like once you open internet, instead of wasting time on other things, you can learn a lot in this. So in the course of preparing these slides, when I came across like one of the presentator like mentioning once a baby is born, the whole genetic code of that baby is also produced from the sample of it, and the baby will carry that particular code of its own, maybe in a chip or whatever. And whenever in future, when something is required, the baby or that particular individual can have the precision medicine. Yeah, there is a lot of time. I mean, takes time, uh, but it can be a future. So I mean to say, like you have a lot of scope here. You have a lot of opportunities where you can. Uh, this is a great fusion of pharmaceutical sciences and uh, bioinformatics. When somebody asks me a question like, "What is your background?" and I say, "B pharmacy," in bioinformatics, people say, "Oh, there are a whole lot different areas." But I say, "No, they are together." In fact, I say that I'm more applied there than anybody. After all, bioinformatics talks with biology, bio life, and we are there to give a quality life. So now databases plays a vital role in bioinformatics. There are different types of databases. Um, you being from pharmacy, um, as I said, databases, you have both sequences and structural databases. Um, so each data which are designed, there are databases across the globe, or I mean, evolved or created based on the research of that particular institutions. So mostly we have NCBI. You can see down at the GenBank hosted. It's called National Center for Biotechnology Information. So you see NCBI. You have it's a umbrella of all other uh, databases, and you also have European based. It's called EMBL, European Nucleotide Archive, or sorry, European Molecular Biology Laboratory. So, and you also have coming up. They are already there, and there are many huge. So you can see like databases based on RNA, based on DNA, based on proteins, and based on their sequences, based on their structure, based on their results after the analysis, based on pathways, and based on like the similarity search or patterns after working with all the sequences. So, uh, yeah, all these databases are free. Anyone can access, can download, anyone can work, and all these databases, the data is free, available. Whatever you take, whatever you work with, whatever results you get, you can publish and you can refer the work from these databases where you got. You have the list of references for each. Say, for example, you take insulin from its data, a DNA sequence or RNA sequence or its protein sequence. You go through it, you have the whole sequence, you have the structure of it, you have um, the explanation of the branches or whatever it is, the part. You have a big annotation of the whole insulin entry itself in a database. So in the end, you have the references who design or who worked with all that particular of that particular entry. So as a student or even you can take this and work on it and publish it and refer because they are all free. And You also have chemical databases. As I said, it's all with the uh, small molecules and chemical compounds and its information and its own structures which are being designed. So coming to biological databases, you have primary databases. It's the very first data raised from the experiments. And secondary databases are, as I just discussed a few minutes back, uh, it is the data erased after working on the primary data. So people are working, and I will come back to this later. And DNA, as I said, you have GenBank designed by NCBI, you have EMBL, yeah, European Nucleotide Archive, ENA, and you have DDBJ designed by DNA Data Bank of Japan, designed by National Center for Genetics. But you have, these are just three which I listed for you. This is the same thing again. And you have protein sequence database like UniProt, SwissProt. You have thousands and thousands of sequences in this protein sequence databases. And the next part is like protein structure. Protein structural databases are a little different. They also have sequences, but most importantly, you have the structure of your protein. And uh, I just want to show a glimpse. Say, for example, I just copied this a couple of hours back. I don't know whether it is visible for you properly because I reduced the size of it. You have Uniprot um, and SwissProt, Tremble, TR-EMBL, 562,253 sequences which are annotated and reviewed. Okay, these are just protein sequences. But when you look down in the same picture, uh, sorry, we have TR-EMBL. You have quite a bit. It is a 9-digit number. So you have so many protein sequences which are automated but not reviewed. So when you come down to PDB data bank, you can check. There's a number 164,840. It is 1,64,840. These are the protein structures available in PDB. So when you compare with the sequences, there is very protein structures which are being uh, available in the database. So there is a lot of scope to work with protein structures alone. So, I mean, not necessarily in vitro. As I said, you can also work with in silico, modeling a protein of your sequence whose structure is unknown, but with the existing sequences which are already there and checking the similarity of those particular expressing portions, regions or genes or whatever, and try to model a protein based on the protein structure of that particular those. So there is a large scope here because unless you have a protein target, you can't design a drug molecule or a lead compound. So coming to chemical databases, um, you have many chemical databases. I just list a few. It's also coming from NCBI, and you have Drug Bank, you have ChemSpider, you have EMBL's database, you have KEG ligand, where you can screen, you can do virtual screening of compounds. yeah, you can just go through all this to get your information for your chemical compound. And I have listed a few softwares. Like you have a lot of freeware, but I just listed one here, ChemSketch from ACD Labs. You can draw your own molecule. You can design your own molecule based on the active site. And once you draw, and one more thing, like I forgot to mention, see, like most of you know, file, every file which we create or try to save in computers, say for example, document, you have a file extension, right? So it has a format. The same way, um, coming to uh, mathematics, here also in databases or what, you have some file formats. When you draw a structure in ChemSketch, you save it in a particular file format which is compatible with that particular other software or other database you're working with. So yeah, I just wanted to add that part. Then you have many commercial softwares. So there are a lot of companies working. There are a lot of pharmaceutical companies. I work in Thermo Fisher Scientific, and I was shocked to see that Thermo Fisher themselves have a commercial software. I don't know whether it's applied in drug designing, but I saw Thermo Fisher LIMS. So Schrodinger is actually is a huge. It's one company. They bought other existing softwares which I worked with. Accelerates. Accelerates is a company. They had Insight 2 for modeling, Series 2 for chemistry modeling, and many other sequence analysis. It's called GCG, and all these put together, they brought in one suite. It's called Discovery Suite or Discovery Informatics Suite, and that comes under one umbrella called Schrodinger. Schrodinger, they gave academic version of software available for institutions to train students and also publish papers with that. And you also have Autodoc. Autodoc is also a commercial tool where you take your target, your small molecule, do docking and force energies, and see how well the lead molecule is working. And I think this is the last slide. And I'll come back to the questions. The application parts. We have applications, but as I already mentioned, uh, which is very applied to us, is personalized medicine, preventive medicine, gene therapy, and drug development, antibiotics resistance. Uh, yeah, I think these are very much applied, and rest every other area where is mathematics applied. Thank you very much for your patience. I took a little more time than what I was given. I don't know. Uh, I just want to go through the questions. Maybe the faculty or students. If the doing is only one drug, only one for drug design. I don't understand this, but I assume like the question is like, is it like one drug designing at a time or working with different compounds? Actually, it's both. Um, as I already did, you can design one particular. It's a ligand based, and if you're working with a receptor, you have multiple lead. You can't say drug. It's a lead molecule. You are going to screen. And can you explain other methods? Uh, I think I mentioned almost like of the methods here, but only thing is you have to dig deep inside, and it will be the whole course of maybe 1 or 2 years of bioinformatics. Thank you for the appreciation. I'm not very informative, but I try my best to give a glimpse of that where, yeah,
Thank
some
you.
type docking. Is it talking about asking what is docking? I think I mentioned what is docking. Uh,
Yes, thank you, Mr. Jason.
uh, yeah, one last question, Venkat. I'm sorry. Can we
Yeah.
answer this? Can you explain disadvantages? A wonderful question by Shamala.
Oh,
Yeah, the problem with yeah, that's the reason bioinformatics has evolved long back. Actually, there are very few drugs which are launched into market in US alone, not more than 10. When I was studying my masters, I heard just 4 or 5, but I don't know how many are solely designed on bioinformatics. The only problem is working completely with computers. We have a theft problem, you have an integrity problem, you have data reliability problem, and so on. So, so something has to be optimized and something has to be refined so that the reliabilities or the integrity or the security is being maintained. And that is possible because working in pharmaceutical industry, when we work with computers, like we use SAP for storage or retrieval or yeah, whatever it is. Even this computer or a software is being validated. So something that I'm just guessing. Uh, I think I answered the questions which are being posted. Oh, sorry. I'm not watching the comments because I'm in a different tab and the comments are in different tab. Uh, Maybe you can email your question. My email ID is ubjason@gmail.com. Thank you, guys. Bye.
yes, thank you, Mr. Jason. So you have given very explanative. So in this regard, that drug discovery, lead molecules, target molecules, you have given differentiation between the lead molecules, drug molecules, and candidate drug molecules. So that is also very nice. And also you move to the drug discovery, receptors, CAD, bioinformatics. So you covered all these topics. So it is very good enough to the students. Within a span of time, you cover all these subjects in a span of time. So I thank you very much
You're welcome.
being part of our online this type of webinars. And also, I have few more questions from the students community. So
Mhm.
they have given two more questions also in the chat.
Yeah.
So one V Sai, he asked one question: which type of receptors based on drugs? So I think he typed which type of receptors based on drugs. That means he's asking based on drugs we can select the receptor like that. Whether I don't know, he asked like this:
Yeah,
which
yeah,
type
yeah.
of receptor based on drugs?
Yeah, I think I covered a little bit about this question. Nothing wrong. I will repeat it again. So
Yes,
when you're thinking or looking for a disease, our drug molecule, when it is going in an individual, it has a target. So that target is mostly a protein. So this particular protein, which is causing the that is our target. We either design a drug candidate or a lead based on the target, or you already have a most active ingredient, maybe naturally or whatever way, and then try to see where it can work with another its most potential target, or a receptor. Receptor is nothing but your protein structure. So it is vice versa. I
Yes.
hope I got my answer.
Yes, sir. Yes. And also one more question is, Mr. Jason. So what is the difference between the lead compound or active ligand? Is there any difference?
Oh, this is from a student.
Yeah, and this is not a from student. This is from faculty.
Oh, okay, that's fine. Uh, can you come again once again?
Is there any difference between the lead compound or active ligand?
Yeah,
And active.
yeah, it's a wonderful question. Maybe even students is they get it's
Yes.
like an information. So when you talk about ligand and what are the two words I forgot again? Sorry.
Lead compound, and active ligand.
Okay. So lead compound or ligand. Ligand is nothing but, as I said, which binds, right? So whatever it is, either of them, unless you a functional part of it, it's called pharmacophore. So unless you have some ions, ionize it, you cannot have a chemical bond. So once you have that, it, that is what it is showing its action. So ligand itself is active actually. So without that, we don't need a compound. We may have n number of chemical compounds, but only which are active, which are based on our target, that is our interest.
Yeah. Thank you. And one more question is from, I think, is the impact of bioinformatics in the development of vaccine. What is the impact of bioinformatics in the development of vaccine?
Wonderful question. Actually, yeah, vaccine development is huge. As you know, vaccine is not a drug. Vaccine is disease itself. So that is our target anyway. That is our protein of interest, and you have pharmaceutical companies working with. Just a moment, my battery is dying. And, oh, sorry about it. Okay, so this is very much applied with bioinformatics because it can speed up the process, and it's nothing but your protein sequence or protein structure again, as I said. So um, when they're working with the most potential targets or working with that particular sequence, yeah, bioinformatics is involved working with the sequences, working with the structure, and so on, and speed up the whole process of vaccine production.
Yes. And one more question.
Mhm.
Is there any homeopathic medicine we can prepare by the bioinformatics? From Pedraidu, he asked the question.
Yeah,
Homeopathic medicines we can prepare by bioinformatics.
yes, actually, uh, that's a wonderful question because when I worked in Chennai, my homeo doctor who also speaking Telugu, but he's from Karnataka. He like we had a discussion. I was like I went there as a like patient or myself or for my family, but he was asking me like,
can we collaborate and work? But that time I was very new, but it never happened again anyways. But this question is new because when it comes to like homeopathy too, you also have chemicals there. You have like they use mother tinctures and you have active ingredients there. You can extract those ingredients, you can elucidate those structures, and based on that also you can make a therapy. But homeopathy is like, see, the I can only give the half answer because every therapy has its own delivery system. Say, for example, pharmacy, it has an own drug delivery system. Even within pharmacy, you have different types again. So homeopathy itself, it has system. So its delivery system, its dosage forms are different. But maybe the one who asked the question, uh, either you have two things, like you can help in homeopathy based on the bioinformatics, working with its chemical information, or you can take that and based on that also maybe we can see a potential candidate on the basis of allopathy.
Yes, Pedraidu, he has given an answer also. Yes, correct, sir. Drug delivery system. Like he commented.
Yes, yes. Oh, is a faculty or a student?
Student, second year.
Oh, B Form.
B Farm second year.
Oh, with the
He's from rural, but he's very intelligent.
So wonderful question. Actually, the only problem with homeopathy.
I just want to add one more point. The reliability of homeopathy is very less. The problem is like the allopathy doesn't accept. That's the, that is where actually this particular homeo doctor asked me in Chennai. Like until we have proofs, right? That is what science is. Until you uh, work with it and prove it, you can't get reliability, right? So that is what is lacking in homeo. And if somebody is working with those actives and trying to get and use the bioinformatics, it will be really wonderful. It's a great thing.
Yes, thank you very much, Jason, for your answers. Very good answers. Also, last but not least, this
I
is
don't know
a
whether they are very good or not.
yes, because students are asking questions, no?
Yeah,
So,
yeah, yeah.
So that we can guess. So what? Because this is very typical content. So even though this CAD technology and drug discovery, this is very good typical content. So students, they have to understand first of all what you are saying.
Yeah, yeah.
And also, what are the opportunities for pharmacists in Canada?
Yeah, I think you asked the right person, and I'm going to answer to the right people.
uh, is there any way I can also
This private,
Yes.
I can. Oh yeah, I just want to give my email ID to all students. I just want to say my students, but
Yes.
anyway, yeah, my students. Okay, that's my email ID. Uh, yeah, coming to my friend's question. Somebody has a question. Sorry. Okay, protein target test.
Jahnavi Sri.
Yeah,
She's
Jahnavi,
asked.
student or faculty?
I don't know. She's from outside.
Oh, okay, okay, okay. You have problems. That is what the toxicological studies is.
so until it reaches the target, drug cannot stay as such because it is highly active. So it can bind any other source, or uh, it may not reach to its destiny. So that is where pharmaceutical sciences matter. You have dissolution studies, you have a lot of optimization. Once the drug candidate is found, you have other things where bioinformatician cannot do. Okay, so it's a good question. To tell you, like all these students or faculties, we have questions. Maybe this is your area of interest, or if it is not so, maybe you can, that will be your most probable area of interest. You can work in some projects, research. Yeah,
Uh,
coming
yeah,
to my friend Venkat's question. Uh, when I came here, I myself I've gone to 1 year program called uh, PG Diploma in Pharmaceutical Research and Development Technology, and I became a student after a long time. So, and then I finished my program, and then I got into Apotex in production operations, and now I'm in Thermo Fisher. And I see a lot of my colleagues; they are all young, like some of you, uh, like students who finished their B Form and maybe even M Form, and they came here on student visa. And some, I have my colleague Ravi. He's from MA College, and he actually posted the poster made by Venkat yesterday, and I was shocked. And he said, I got this poster from my group from MA College. So he himself is from pharmacy background. So he came on PR. So the scope is like you can come on student visa, take a program, maybe 1 year or 2 years or whatever. And once you finish that, as you're all in the related field, pharmaceutical companies, they're taking freshers more. And you have, as I said, there are two different types of pharmaceutical companies. One, the regular allopathic medicines, like orals or parentals, whatever. But there are other set of pharmaceutical companies of biopharmaceuticals. Like for example, Sanofi Pasteur, they work making vaccines or whatever based on recombinant DNA technology. So it's like more of biotechnology. But once uh, um, uh, you get the product where it is used for the treatment or as a vaccine, whatever. So as a large, you have two, and in both areas, you have a scope of jobs. And yeah, coming to Canada, yeah, there's one thing like once you finish your 1 year program, you get 1 year work permit, or if you take 2 years program, you get 3 years work permit. And once you get finish your college, you can get into a job. And if it is, there are different grades, at least a good, at least a manufacturing side or whatever, you get 1 year um, experience, you can apply for a PR and you can be a permanent resident of Canada. And yeah, you also have universities which are um, top in the world. And number one is UFT, and many other universities, they work with drug designing, and even in the pharmaceutical area, like working with compression or pre-formulation studies, formulation studies, and so on. And you also have a lot of CROs too. You have clinical studies going on here. And if you wish to know like how is the job market, as I said, just Google Indeed.ca. You have all the jobs there.
yes. Thank you, Professor Jason. So there are so many students are asking questions also. I think it is already 8 o'clock. So yeah, I am very thankful to you.
Any
And,
more? Oh, okay. Okay. If you have any important question, I
one Pujita is asking, I have doubt that many say that RMP doctors used to prescribe only painkillers and antibiotics. Heavy usage of these kind of drugs is not recommended, so it is correct.
yeah, exactly. You're right. Going to the root problem is important than just going with the symptoms.
Yeah, going with the symptoms and prescribing the drug is important immediately what a patient requires, but gradually one has to know what is the very root problem of it, and that cannot be done by an RMP doctor. And Pujita, you asked me that RMP doctors are prescribing, but this is also prescribed by any other doctor too. Uh, Maybe first it is required, but a thorough examination is important than just working with the symptoms. You're welcome. You're welcome, everyone, and thank you for the patience.
Yes. Thank you very much, Professor Jason. Most of the students are asking that feedback link. So that feedback form will be provided in description page.
Yeah, somebody is asking any opportunities for Farm D. Swati.
Yes, in Canada.
Farm D, I think I don't know much about Farm D myself because I'm cut off from
Pharmacy colleges long time back. Uh, can you tell Venkat what Form D has?
like.
So this is a doctor of pharmacy, particularly related to the clinical pharmacy.
Okay, okay. So I think Swati, if you're like Farm
D, you're mostly like you have two things like you can get into retail or into hospital, and mostly most of the pharmacy students who come here they go into retail, but once you come here you have to pass the regulatory exams, clear them, and you have to be licensed, and then you are a pharmacist. And pharmacist in Canada, you are paid well. It's not like India, and you're respected more. When somebody steps into a pharmacy, you can see the difference, like other assistant technicians and how the people respect pharmacists. Like just like a doctor in a clinic, pharmacist in a pharmacy. So that's one thing, retail, and then you also have opportunity to work with hospitals, but there it is most basically on community. So there you need other skills, but everything is possible. Like Form D, you have put full potential to clear the exams and be a licensed pharmacist and work, and you're paid well and you're respected well too.
Yes. Thank you, Jason. So on behalf of St. Mary's Group of Institutions and from my student side, so thank you for your nice session and queries also. Thank you very much.
and one last thing, like all students and faculty, stay safe, be careful, and maintain social distance and try to prevent this COVID-19.
Take care, guys. Bye.
Yes. I thank to each and everyone for participants. I thank to my
I think
students and
Pavani is asking my email ID, but I think I already text my email. You can look into the chat.
yeah. So okay.
Okay then. Bye.
Thank
See
you.
you.
Bye.
See you, Venkat.
Thank you.
Thank you very much.
Thank you.
Yeah. Bye.
Thank
Bye.
you.